Hi, and thank you for inviting me to speak in BioConference Live. This is David Symer. I'm going to tell you about some of our recent work where we have been studying the effects of human papillomavirus in human cancers. We've taken a multidisciplinary approach to try to understand the biological and genetic impacts. So I wanted to start first by thanking my great team of collaborators and colleagues. They include members of my laboratory and Maura Gillison's laboratory at Ohio State, along with Ted Technos. In particular, I wanted to acknowledge and thank the tremendous bioinformatic work of Keiko Akagi, and as well to thank the wonderful work, molecular work of Jingfeng Li in the lab. As well, we have collaborators at the National Cancer Institute and Thomas Reed's laboratory, and at Mass General at Harvard University. I'm sorry it's overwritten there, but from James Rocco's lab, and funding is indicated there below. So what's known about human papillomavirus? Well, they've been known to cause human cancers for several decades now. These are small circular DNA viruses. Humans are the only host of them. There are over 130 unique types of HPV, and they infect both cutaneous and mucosal sites. There are high-risk and low-risk types of virus. So we're going to focus on impacts in genomes of cancer cells, and so we're studying high-risk HPV types. The genomic structure of HPV is shown here. There are early and late genes indicated. The famous oncogenes are E6 and E7 over here. I will try to circle them for you. There they are. As well, there are late genes, and then there is an upstream regulatory region, which is a promoter for the circular viral genome. E6 and E7 are oncoproteins, and they disrupt the famous tumor suppressor genes P53 and PRB in the host cells. And they disrupt their function, leading to cancer. So when we started our project, what did we already know about impacts of HPV integration in human cancers? Frequently, the virus is thought to be episomal in head and neck cancers and in cervical precancer lesions. Integration has been correlated in cervical dysplasia with severity. Integration seems to stabilize E6 and E7 transcript stability. And integration is thought to disrupt the viral gene E2. However, to date, there have been minimal functional consequences known of viral integration. So that's what we wanted to study in our project here. So our goals were to characterize the viral genome in cancer cells, and as well to understand the HPV transcriptome by conducting RNA-seq. And in particular, we wanted to look at the genomic integration events of the virus in host genomes. We wanted to investigate potential interactions between the viral and host genomes in cancers, and wanted to find any potential functional consequences of viral integration. So in our initial project, we started with 10 well-established cancer cell lines, and additionally, we had 2 primary head and neck tumors available. We used the Illumina Genome Network on the HiSeq 2000 sequencing platform. We conducted 100 base pair paired end read sequencing, where the average library fragment size for our whole genome shotgun approach was 300 bases. And we obtained per genome about 100 gigabases of coverage at 30x at least coverage across the genome. We used Illumina's pipeline to align originally the whole genome sequence data, and then we mapped viral insertional junctions using a custom pipeline that Keiko Akagi developed using BWA and GSNAP software. In addition, we characterized and confirmed integration sites using polymerase chain reaction and Sanger sequencing. And we had to reconstruct and confirm these complex genomic structures that I'm about to tell you about. So one of the first things that we did in this study was to characterize mutations in well-known cancer-causing genes that have already been found to be players in human cancer formation. And the list is shown here. We found typically a similar number of single nucleotide polymorphisms and insertional deletional polymorphisms in each cancer genome. And as well, 9 of the 12 samples that we were studying was HPV positive. 3 of them were HPV negative, and in each of those they had a concomitant P53 mutation. So typically one or the other mutation or the viral presence was part of each genome. In addition, we saw these mutations in other well-known cancer-causing genes, including CDKN2A and others as shown here in this table. This was recently published in Genome Research, and the reference is shown here. So when we looked at the viral genome in each of these cancers, the type was typically the high-risk type HPV-16. One of the primary tumors, which we call tumor A, an oral cancer, had HPV-18. They had typically a range of copy numbers per genome, which we characterized using quantitative PCR, ranging from 1 up to several hundred, as you can see, up to over 800 different copies of the viral genome in each cancer cell that we studied. So when we mapped the coverage of viral whole genome sequencing to the viral reference genome, shown at the bottom of this figure, we saw a wide range of coverage of the virus, as you can see. This is using the Integrated Genome Viewer from the Broad Institute, IGV. And what you can see in these blue histograms is the depth of coverage of the whole genome sequencing across the viral genome. And what you can see in particular here is that the E6 and E7 genes were present in every single cancer cell line or tumor that we studied. By contrast, some of the parts of the viral genome were completely absent from the viral genome, and now I'm going to circle some examples of that here for the top cell line, which is HMS001. You can see that the viral genome was not present at the stretching from E1 up through about L2 of the viral genome. The red histograms indicate the viral host breakpoints, and what you can see is that the virus integrated into the host genome in each of these cells samples. As well, when we map these insertional breakpoints across the viral genome, what you can see is that we were able to verify virtually all of the insertional breakpoints that we identified. We verified them using PCR. And the size of these circles in this histogram are indicating the number of whole genome reads that we use to validate each insertional breakpoint. So this refuted a conventionally held dogma, which is that the virus has insertional breakpoints that occur in particular spots of the virus genome. And instead, what we observed is that the insertional breakpoints were distributed all across the viral genome. Now, when we used RNA-seq to analyze the HPV transcriptome in cancer cell lines and tumors, what we saw is that every single cell line or tumor that we studied had HPV E6 and E7 transcribed. And these sashimi plots, as they're so-called, from RNA-seq data indicate that the RNA-seq reads were mapping to the E6 and E7 genes again. So I'm going to circle that here. And what you can see is that E6 and E7 were always present in all of these cancer samples. By contrast, when we looked at the copy numbers of the host genome in HPV positive versus HPV negative cancers, we saw characteristic gains and losses of certain chromosomal arms. So this has already been quite well known in the field of HPV cancer formation, but very frequently chromosome 3P was lost and chromosome 3Q, the long arm of chromosome 3, was gained in most of the samples, whether they were HPV positive or negative. And this has been confirmed by other groups over the last many years using a variety of methods. When we plotted our data using surface plots, this is looking at one of the cell lines that we characterized. What we saw, very consistent with what I just showed you a few slides back, was that the HPV genome was partially represented So that in the orange histogram here going around the viral genome in a circle, what you can see is that the coverage of the whole genome reads indicated that certain parts of the viral genome were present, but other parts were absent. And in particular, breakpoints going into the host genome are indicated in these red dots here. And the virus integrated in a very characteristic fashion, as I'm about to tell you. When we look at the karyotype of the whole host genome on the right surface plot, what you can see in the red arcs is the chromosomal translocations that we had high confidence to support. The histograms going around the circle indicate gains and losses of chromosome arms that we identified using the whole genome sequencing. So let's zoom in and look at the impact of virus insertions at a couple of locations in a couple of our cancer samples. So again, these are IGV views. These graphs are indicating depth of coverage of our whole genome sequencing read. And as a comparison, we plotted the coverage for a control normal cell in yellow. In blue was the cancer cell line that's indicated. So at the top is the well-known cervical cancer cell CHA. At the bottom is a head and neck cancer cell called HMS001. And what you can see in these histograms is that right around the sites of viral integration are these very large gains and losses of coverage of the sequencing, indicating that there is focal amplifications of the host genome right near the viral insertion. These histograms in red indicate breakpoints between the virus and the host genome. I'm circling a couple of them. And you can see that the host genome and the virus insertion right into the host genome immediately flanks these sites of host copy number variations and rearrangements. So we saw the same kind of pattern again and again as we looked across the different cancers that we studied. And we came up with a model to explain how these complex genomic structures could be arising and how they could be explained. This is a model that we call looping, and what we're indicating here at the top in the shadowy bluish area is a histogram indicating, again, the depth of coverage of the whole genome sequencing. These red arcs are connecting the host-host breakpoints that we observed. And then in black, we saw deletions of host genome. And then these red arrows that are indicated in this graph indicate viral insertions themselves. And so what we saw was a complex structure where the virus insertions went right into the host genome and mediated somehow these complex host genome amplifications and rearrangements right around the viral insertion sites. So this model we call looping, and what we infer is that when the virus inserts into the host genome, perhaps its origin of replication mediates replication of DNA strands, which loop and transiently form these loop structures, which then resolve into concatemer arrays of the virus and the host genomes together. So I'll give you a couple more examples of this. Here's another cell line, a cancer cell line, again called HMS001. We were just talking about this. HPV happened to land into a transcription factor called EYA2. And what you can see is right around the sites of viral insertion were host amplification and rearrangement. And the virus was right in the middle of all of this action. Again, our model for looping of how this happened is indicated at the bottom. And if you want to have more detail about our model, certainly please look up our recently published paper in Genome Research. It explains it in more detail than I have time to tell you about here today. So here's the model. Here at the top is a host genome sequence, and you can see segments marked A through F. At the top is your favorite gene, that's YFG up at the top with its exons and introns indicated. And in red is indicated the HPV viral genome. And again, this is our model to explain these structures that we observe in the cancer cells long after the act of viral integration happened. And so what we're saying is that transiently the virus inserts, and then some focal genomic replication happens with rearrangements and amplification, And the end result is concatemerized arrays of these complex copy number variations and rearrangements that seem to have the virus inserted right into the middle of them. Again, I'll give you a couple more examples of this. So here's several more cancers that we studied, and you can see sometimes that the amplifications of the host genome around the viral insertions are massive. So in these blue histograms at the top are the amount of depth of coverage of the host genome. What you can see is that in some cases the amplification was up over 50-fold or even close to 100-fold amplification locally in copy number variants. In red again are the viral breakpoints where the virus inserted into the host, and again we see the virus and the host genomic instability go together in all of these kinds of cancers that we studied. We also saw examples of host genome deletions mediated by the virus insertional process. So here in yellow again at the top is a histogram indicating depth of coverage of a control sample. In blue is our cancer cell line SCC2. And what you can see is that there's a big gap right here in the middle, which I'm circling right now, which was presumably mediated by the viral insertional process, so that when the virus went in, the host rearrangements happened, and this chunk of the host genome was deleted in the process of the viral insertion. So we saw some examples of cancer cells that had lots of instability and complex integration events. Here are some circus plots indicating the viral genome on the left and the host genome on the right in Kasky cells, another well-established cancer cell line. And again, we indicated host breakpoints and viral insertional breakpoints in these dots on the circus plot on the left. As well, there's all of these chromosomal translocations that you can see with these massive number of arcing arcs around the circus plot. On the right is the host genome, and you can see the karyotype of the human genome going around the circle from 1 to 22, X and Y. And then as well, you can see the depth of coverage of each chromosome, and again, where are the insertional breakpoints. So we were a little concerned about using just the whole genome sequencing data to understand such complicated events. And so we turned to our colleague and collaborator, Thomas Reed at the NCI, who's well-known cytogeneticist, and he used beautiful fluorescence immunohistochemistry, in situ hybridization, as well as spectral karyotyping to confirm that the HPV insertional events correlated with some of these chromosomal translocations and breakpoints and so on. And so again, this is presented in our recent genome research paper, but you can look and see that where are the red dots here on the bottom left, which I'm circling now, those are the HPV insertional sites, and they seem to correlate, at least in many cases, to chromosomal rearrangements that you can see from the SKY results here in the center. So very interesting, and in general, the FISH and SKY confirmed very nicely what we found with the whole genome sequencing analysis. So we also saw evidence for chromosomal translocations that were mediated by HPV insertional events. This was looking at a primary tumor. Again, it's presented in our genome research paper. This is a tumor that we call tumor B. What you can see is that there were viral breakpoints on chromosome 11 and chromosome 8 here on the top and bottom, and they are bridged together by this kind of a looping event where there are host genome amplifications and deletions and translocation all together, connecting these two chromosomes in the tumor. So this is just more evidence for the same phenomenon. It's presented in that paper, so I'm going to go quickly over it because time is limited. And again, we found many cases where there is quite a lot of evidence for complex structural rearrangements around HPV insertional sites. This is now focusing on another primary tumor, which we call tumor A, and we found HPV-18 in this tumor, not HPV-16. And again, the same story, where you saw huge amplifications and rearrangements of the host genome, indicated up here at the top, connecting parts of the genome that are normally not contiguous. And then we tried to resolve these structures as indicated at the bottom. Again, consistent with our looping model, some of these rearrangements happened 15 to 30 times, connected by viral insertion sites. We characterized across this panel the extent of hyperamplification and the extent of structural variation that we observed. And what we saw was that in a local window around the virus, there was a very high frequency of both hyperamplification as well as structural variation correlating with HPV insertion sites in each and every sample, with very few exceptions. So in summary, what I'm trying to say here is that when we add up all of these insertional events in the cancer cell lines and tumors that we studied in our first project, we see viral insertions that are really hitting across the human genome in a wide range of locations. It seems to be fairly random as a process of where does the virus insert. But then in any given sample, we see again and again evidence for local insertional events that are clustered, and as well bridging these local amplifications and rearrangements. So when we plot all of the insertions across the human genome from all of the tumors that we've looked at, it looks in some level like they're clustering in hotspots, but these are not from different tumors. These are all, each of these clusters is occurring in one cancer sample. And so what we're seeing is that when the virus inserts, it seems to go in, and then a lot of rearrangements happen such that the viral copies increase over time right at the site in the locus of the initial insertional events. So last year, a beautiful paper appeared in Nature from Jay Shanduri's lab at University of Washington in Nature, indicating that they had used a range of genomic methods to study the HeLa genome, the well-known cancer cell line HeLa cells. And they presented evidence really supporting the same phenomenon that I'm talking about, which we call HPV looping. So you can take a look at their paper and find really good support with haplotype resolution, indicating that, again, these structural rearrangements blank and are right around HPV insertional sites. We turned to transcriptome analysis using RNA-seq, and we prepared a range of different libraries. This was work largely done by Jingfeng Li in the lab. Beautiful work where he prepared a series of libraries to characterize the transcriptome in all of our cancer cells. Then the alignments were performed by Keiko Akagi using Top Hat or GSNAP software, where she aligned each of these transcriptomes to the human genome reference genome, HG19, together with the HPV reference genomes. And then she detected chimeric transcripts using a software package called Hydra. And in this analysis, we found a lot of evidence for fusion transcripts bridging between viral insertions and then host genome flanking nearby in many or most of our cancer samples. And you can see a predominant gene that was hit in each of these. Many of them appear to be well-known contributors to cancer, such as TP63, which I'll tell a little bit more about in a few moments. So this was an example in the cell line SEC090 on chromosome 9, where we found amplification and rearrangements around a transcription factor called FOXY1. Our RNA-seq data indicated that FOXY1's expression was massively upregulated in the same cell line in comparison to all the other cells, which don't have HPV integration at the same site. And what you can see from a heat map analysis is that FOXY1 and its neighbor on chromosome 9 are dramatically upregulated, specifically in the cell line that has this looping event of HPV in the neighborhood. Another example of a genetic impact of the virus is shown here, where in the cell line called SEC47, we saw insertional breakpoints at a gene called TP63, part of the very well-known P53 family, where the virus had inserted right into the gene. And you can see, again, massive upregulation of, or copy number change, I should say, of the genome. And then what we found by looking at the RNA-seq data and using race analysis is a large number of orphan transcripts that were fusion transcripts expressed from the virus downstream of the viral insertional site, so that we saw a range of splice variants that were 3' end truncated transcripts of P63, as shown here. And when we looked on northern analysis, we saw the predominant band on northern was shortened in the cell line SEC47 in comparison to control samples shown here. And western blotting showed that carboxy-terminal truncated protein was expressed from this truncated transcript. So this is an example of HPV breaking a gene and really disrupting it in the cancer cell. Now, more recently, we turned to study a collection of primary tumors to understand whether or not the same kind of events can be happening broadly across human cancers that are associated with HPV. And I should say that HPV is associated with about 5% of all human cancers, and in certain anatomical sites that rate is increasing, and that includes head and neck cancers. So when we did whole genome sequencing in a panel of human cancers, we found, again, as I said before, a range of insertional breakpoints connecting the viral genome at the top to the host genome at the bottom across a range of sites in the different samples. As well, we recently have conducted long-range sequencing using a novel platform that Illumina provides. And this confirms some of these complex structural variants that we've identified in our samples. So this is one of the primary tumors that we've been studying. And again, at the top, you can see really strong evidence for a looping event. I've shown you this several times, so this should look familiar. But what you can see is amplification of the host genome. It ranges up to maybe 10-fold amplification of the host genome. And then as well, we see evidence for breakpoints that connect the different parts of this rearranged structure. And these were confirmed using this long-range platform from Illumina. So the way that this works is Illumina receives whole genome DNA, and they fragment the DNA to approximately 10 kilobase fragments, and then they fragment those 10 kb chunks and barcode them, sequence them all, and then recombine them into 10 kb contigs, which are these long reads shown at the bottom, which I'm circling now. And what you can see is that these long reads faithfully confirmed our structures, our models, of how HPV inserted into the host genome and bridged these discontinuous parts of the genome. And so each of these colored lines here, purple and orange and red and so on, indicates a different long read that captured different parts of these insertional breakpoints that we identified. And so what this indicates is that our model seems to be correct so far in terms of these complex genomic structural variants and these structures that we're observing. And we think that long-read sequencing can certainly help in the future to resolve additional complicated structures. So one thing that we observed in this case that I'm showing in this figure is that actually the virus structure itself was different in different copies of the insertion. So at the top is an intact piece of the viral genome, and at the bottom is a piece of the viral genome that lacked an internal, approximately 50 or 75 base pair fragment of the virus that is the little white gap here at the bottom of this figure. And the long-read sequencing was able to resolve this in a way that we would not have been able to see it previously. So we're hoping that the long-read sequencing will really shed a lot of additional light on the structures that we see very commonly in HPV-associated cancers. So just in conclusion, we're using a range of molecular methods, including whole genome sequencing, as well as this long-read sequencing, as well as FISH and SKY and other methods to characterize HPV insertions in human cancers. We found a spectrum of different mutations in the cancers, as was expected, and we saw frequently a loss of E2 of the virus. We see widespread integration sites without really evidence for hotspots of viral insertion we do see local clustering of these events in cancers so that in one given tumor, we see again and again the virus is present in the neighborhood where it has landed in different structures. But across different tumors, we don't see the same sites being used exactly again and again. We see evidence with RNA-seq of viral host fusion transcripts, and we very consistently observe expression of the viral oncogenes E6 and E7. So we identified, in summary, we identified that very commonly in human cancers, HPV is associated with focal genomic instability, and we proposed a looping model as the basis for this instability. And as I said before, if you want to read more about it, please turn to our recent paper in Genome Research. We found that the virus disrupts host genes by a variety of mechanisms. They can include amplifications, deletions, chromosomal translocations, antisense transcripts, gene breaking. And then, as I said just at the end here, we're using currently long read sequencing to resolve and confirm some of these complex structural variants that are associated with HPV insertions. So I'm just going to end with my acknowledgement slide again. I really appreciate the contributions of our fabulous team that we have listed here. I'm going to hopefully turn on the question and answer section here. So if you have questions, please enter them in and I will see if I can address any of them. I see there are no questions at the moment, so I'm just going to wait for a moment and see if you have any questions. I would like in the meantime also to thank Bioconference Live for hosting us. This is, I think, an interesting and great new way to present data. And so if any of you have any questions, please enter them in. I'm going to wait a couple moments here. Okay, so there is a question, comes from NIH, and the question is, does this contribute to chromothripsis? And it's a great question. We think that it's distinct. We think that this looping process may be distinct from mechanisms of chromothripsis for a variety of reasons, and the main one is that these events appear to have some level of organization to them. So they are recurrent, and we see concatemers of the host genome that are arrayed head to tail, and inverted, and so on, that are really organized. By contrast, my understanding of chromothripsis is that it really is kind of a random shattering locally of a chromosome, so that chunks of chromosomal fragments are stitched together in a connectivity pattern that's we can't discern a pattern. It's random, and so that's one big difference, I think, between them. But it's an interesting question. Maybe the phenomenon of chromothripsis has some relationship to origin of replication or something that's that we don't know about at the moment. So a very interesting question. So there's another question, and that is, are the HPV subtypes that are not high risk or cancerous unable to cause genomic amplifications and rearrangements? And that's an interesting question, and I have to say I don't know the answer to that. I have a sense that the low-risk types are likely not integrated, but I believe that they're maintained episomally, and they don't cause cancer, and so their persistence is unlikely. And I'm not sure that that's been very well studied, but that is an interesting question, and I can certainly follow up further offline with you if you want to email me, and we can discuss further. So there's another question, again from NIH, and it's, can you see this in cancers other than those traditionally associated with human papillomavirus? And the answer is, there are interesting similarities with other viral insertional events that happen in other types of cancer that are not HPV related. And a famous example is hepatitis B virus insertions that occur in liver cancers. And they seem to be, there are many similarities really between the focal genomic instability that we see with HPV and what others have seen recently with HBV. So I don't see any more questions. I'm going to wait just a more moment or two, and then we're going to sign off. So thank you all so much for attending. I really appreciate it. And if you have any further questions, just send an email. We're happy to follow up. Thank you.